Thank you for coming to the meeting tonight. It's great to see folks. It looks like there's more people, but maybe because it's a smaller room that we're in. But it's really great to see you. So the meeting is recording. I have my notes. I didn't have them last time. My name is Robert. I'm the president of the PHS. And I just want to make sure that folks can hear me on Zoom real quick. Here. Yes, great, excellent. Tucson is surrounded by herpetological place names in the O'odham language. I work at Tumamakil or Chamamagduag, which means horned lizard mountain. And across the valley are the Catalinas, but they're also known as Babadoag or Toad Mountain. And you can see why. This is the west side of Push Ridge from looking, I think, from Shannon Road to the Catalinas. So this is Babadak, toad sitting. And of course, we are all here on indigenous land, home to very diverse cultures. We have some great merchandise. And if you would like to have some merchandise, you can buy it from Maggie, who's the back of the room here. And our credit card reader is charging right now, but we can accept your credit card information.
Log in.
Okay. So yeah, this is my friend who's wearing our wonderful shirt in New York City. And these are some of our partners that we've worked with. Madrid Discovery Expeditions. We've supported the Mexican Tortoise Project. Fauna del Noroeste, which is a ecological monitoring group in Baja California, and they tend to focus on herps. And of course, we help establish the Cecil Qualby cold-blooded research fund here at the University of Arizona. Second. Right. So this is a new book project. The Snakes of Arizona came out, and the companion volumes are currently being produced. The amphibians and turtles and the lizards. And this website is established to help collect any photos that you'd like to contribute or vouchers, species records. So the Festival of Books is this weekend, and we're still looking for volunteers. We have a pool of volunteers right now, but the more the merrier. And if you're interested in hanging out with children and snakes and turtles and like to share that wonderment with the public, let us know. We'll also be judging projects for the Southern Arizona Research, Science and Engineering Fair, March 4th through 6th. And if you're interested in helping to judge, let me or Maggie know.
Yeah, let me know. The judging will be online. You don't have to go there. If you want to help us judge, I can give you the links. Just so talk to me, so I'll have your email and join us.
All right. We will very soon be determining a date for opening a call for proposals for the Charles Lowe Herpetological Research Fund and the Sonoran Desert Toad Fund. We'll be updating the website and posting the dates very shortly here. And then I think Larry had an announcement, perhaps?
Yeah. So shamelessly, if you want to buy one of my venomous animals of the US and Canada book, I brought a couple of them over here. And those are $45, $5 to go to THS. But that's not why I'm here. So, pre-COVID, I planned to do a field trip, Chuckwallapalooza, which we were supposed to do annually. And went down in a fiery crash due to COVID. So I'm firing it back up again, and tentatively March or April 21 and 22, we're gonna take a trip out to White Tank Mountains, up west of Phoenix. So we don't have the stuff on the website yet, but we'll get that up there and have all the details. You contact me and let me know if you want to do that. And then the other thing is I'm doing a lizard project. I've been doing it for 3 years now. I'm just going to start it up again, looking at desert iguanas and long-nosed leopard lizards. So if anybody's interested in helping at all, come and contact me.
Excellent. If you're a board member, raise your hand. If you have any questions about the THS or joining or wanting to volunteer, just head to one of us. This is our 35th year. If you have any interesting ideas about how to celebrate, let us know. I was thinking today, maybe we issue a special, a second collected papers of the THS. So we have a out of print book, which collected all the best articles from our newsletter from beginning to 1991. And maybe we could do a second one of those perhaps. Is there a particular type of trip you would like to organize? Are you interested in maybe going on a expedition to Sonora, where we could team up with Madrean Discovery Expeditions for an interesting expedition? So let us know. If you don't have a ticket, get one from Karen, who's wearing the turquoise Gila Monster sweatshirt, and you can select one or two things from that table over there. And these are from Cecil Schwalbe, who passed away recently, and there's some goodies there that he would probably like you to have. So they're there. Before I introduce our speaker, I'd like to let you know our upcoming speakers. In March, we'll have Matt Good, who will be talking about 20 plus years of tiger rattlesnake ecology. In April, Annie Peralta and Jorge Valdez, who run Fauno, are going to talk about their organization and their herp projects. In May, Randy Babb is going to talk about introduced exotic amphibians and reptiles of Arizona. In June, Tom Jones is going to give us a historical review of herpetology in Arizona. July is open, so if you want to talk about something, let me know. In August, Taggart Butterfield is going to talk about spotted box turtles. September, Rafael Lara on ecophysiology of native reptiles. And the rest of the months are still open, and I might give a talk as well. I don't know on what yet. I have two things I'm thinking about, so it's going to be a great lineup of speakers, so I hope you can make those. So tonight, we're going to welcome Dean H. Leavitt, and forgive me if I mispronounce your name. He's a professor from San Diego Mesa College who will present on the phylogeography of the Western Banded Gecko, Coleonyx variegatus. The Western Banded Gecko has a large distribution throughout the deserts of the southwestern United States and northwestern Mexico. Up to 7 subspecies have been recognized, including one, which is our mascot, Coleonyx variegatus bogerti. In this talk, he will discuss what genetic data tells us about the distinctiveness of these subspecies and hybrid zone dynamics at one area of secondary contact between distinct lineages. And stop sharing. And I think you can start your share there.
All right. Good afternoon. Can you folks hear me okay?
Yes.
Yeah. Good. Well, good to be here with you this evening. And yeah, I'm fortunate to have the opportunity to talk about one of the things I love the most, which is banded geckos and kind of genetic spatial structure with these creatures. So I've long kind of been interested in deserts and lizards. I grew up in Utah, and I camped a lot growing up, but I would kind of force my parents to go camping in the desert because there you would find lizards. We're going to the mountains. There were fewer of those. So I had a real kind of soft place in my heart for places like, Mojave Desert. And of course, the saguaros of Arizona. I kind of looked at books with admiration and dreaming of when I'd be able to go explore those places. So my first kind of foray into looking at phylogeography focused, I guess you could say, on yuccas, Joshua trees, and the creatures that live under them. I did my master's program at BYU in Utah, and there I worked with Jack Seitz and Keith Crandall and Robert Bezy, looking at essentially phylogeography of Xanthusia, the Xanthusia vigilis group. And our study there was distribution-wide with a focus on Southern California. And kind of after this work or through this work, I became really interested in the biogeography of Baja California. And, a lot of the story of the origin of Xanthusia lineages has to do with the geological formation of Baja California. So I'm in San Diego. I've been in San Diego for years now, which, puts me near the Baja California Peninsula, which I feel, fortunate to be close to. I'd certainly love to visit more often. And as most folks know, it's a desert lover's wonderland. It's not just the lizards and snakes, but the vegetation as well. But it is also about the lizards and snakes, right? If you think about the unique vertebrates of the Baja California Peninsula, I mean, squamate reptiles is the name of the game. There's species found there and nowhere else. Other vertebrate groups from, amphibians to mammals, they don't show the unique diversity patterns in Baja California that squamate reptiles do. So peninsula region of Baja California, at least 170 snakes and lizards, with many of those being endemic to the peninsula. So, coming from having done work on Xanthusia, I was jazzed thinking about continuing work on Baja California. Now, how we think about the origins of Baja California snakes and lizards is primarily due to three folks, I would say. The first of is Jay Savage. He published a biogeographic scenario in 1960, explaining the patterns we see for snakes and lizards in Baja California that was very, let's say, dispersal based and integrated patterns of squamate reptiles with phylogeographic patterns. And at that point, the geologic origin of Baja California was yet to be understood. The plate tectonics had kind of yet to be embraced by geologists and certainly biologists at that point. So, it was sort of ignored. I would say that the next person to really that merits credit for developing models of biogeography for Baja California is Robert Murphy. Published a really fascinating paper in the early 80s. Part of his perspective on Baja California diversity was based on allozyme data that he had collected, and other hypotheses might have been more anatomical or morphological based. But he was the first person that integrated origin of Baja California taxa along with the tectonic origin of Baja California, where it was, over 7 million years ago, it was part of the western coast of Mexico. So you get kind of this comprehensive scenario for the snakes and lizards of Baja California. And the next person that helped kind of push along the development of thinking about Baja California biogeography is Lee Grismer. Of course, he's renowned for his beautiful, amazing book looking at the reptiles and amphibians of Baja California. And he published a nifty paper in 1994. And in this paper, he more or less came up with, let's say, 3 primary groups that different herps could be grouped in based on their biogeographic origin. So some of those species he placed in this group that he called the southern vicariant group. Now, these were essentially species that he hypothesized originated when the Southern Cape region first separated from the Mexican mainland, right? When he hypothesized that to be around 5.5 million years ago, based on the available geological data. The next group of organisms he placed in a group called the northern vicariant group. They also originated via kind of the formation of the northern Baja California Peninsula, and that happened at a younger time than the Southern Cape region. He placed that around 3 million years. You can kind of see that in the figure there on the left. And then there were a number of other groups. I'm just going to kind of call the dispersal group. These are species that essentially entered the Baja California post the formation of the peninsula, anytime after 3 million years, but kind of more or less during the Pleistocene. So these were kind of geologically young species. So this was really nice because it gave explicit hypotheses for the origin of various snake and lizard groups, and it formed a nice model that others were able to kind of follow up on with DNA sequence data. In one of the first papers that was testing biogeographic origins of Baja taxa was Brett Riddle et al.'s paper in the year 2000, where it looked at not just herps, but incorporated some mammals there as well. So there have been a variety of, let's say, genetic tests of Grismer's different groups, right? And some of those, let's say, tests of these hypotheses have held up, looking at the origin of 2 really unique members of Scoloporus, formerly placed in the genus Sator, right? Scoloporus angustus and grandavis, only found in southern Baja California. These were part of Grismer's southern vicariant group. And indeed, when you collect DNA sequence data, reconstruct phylogenies, yeah, you do find that their closest living relative is Scoloporus uniformis in coastal Mexico, south to Guerrero, and that this relationship between the Baja taxa and the mainland Mexico taxa is old, right? So yeah, kind of support for this old vicariant origin or old origin related to the formation of Baja California. And other hypotheses or other tests of these hypotheses have come to different conclusions. Another species that was placed in this southern vicariant group by Grismer was Thamnophis validus in the Cape region. He thought that that had an old southern origin. DNA sequence data, however, show that the Baja California population of Thamnophis validus is geologically young in the Pleistocene, and that it was the result not of the geological formation of Baja California, but rather overwater dispersal from coastal Mexico to the Cape region. So some cool results there as far as having these hypotheses that Murphy and Grismer developed that can be tested with genetic data. Now, my focal taxa for looking at these biogeographic patterns is, of course, the Western Banded Gecko, Coleonyx variegatus. I remember the first time I ever saw one of these. I was in high school in southern Utah by a little town called Laverkin with Breck Bartholomew, and we found one of these, and I was enthralled by this organism. And then years later, starting my master's project, I met up with Bob Bezy in Tucson, and we headed west to California on the 8th, and there in western Arizona off the freeway, we flipped flipped some garbage and I saw a couple geckos, and Bob Bezy saw how excited I was about seeing these things. Are you sure you don't want to switch from Xanthusia to Coleonyx? And well, I stuck with Xanthusia for the time being then, but I did squirrel away as Coleonyx was the next thing that I wanted to study when I was done with night lizard. So anyway, a really elegant lizard and good for testing biogeographic hypotheses, right? Distributed throughout the Sonoran Desert, throughout the Mojave Desert, throughout the Peninsular Desert, which I refer to Baja California as the Peninsular Desert, and they're found throughout coastal California as well. So there are 7 subspecies under the umbrella of Coleonyx variegatus. These were described by famous San Diegan Lawrence Klauber in 1945. I know this map isn't, the clearest, but it shows kind of the distribution of these subspecies. You might be more familiar with maybe a map from the 3rd edition of Stebbins, right? And seeing the different subspecies of Coleonyx variegatus there. One of the subspecies isn't depicted in this map. That's a subspecies Sleveni that's found on a little island in the central Gulf region. But otherwise, we have the subspecies of Coleonyx variegatus represented here. Like Robert mentioned, that includes the mascot of THS, the Tucson banded gecko, right? Now, in Grismer's biogeographic model of 1994, he put Coleonyx variegatus in the northern vipariant group. And so the hypothesis here was that, well, the formation of the northern Gulf of California influenced the phylogeographic or biogeographic structure of Coleonyx variegatus. And he thought there were 2 groups within Coleonyx variegatus: the Abaddii group, which included peninsular subspecies like Coleonyx variegatus peninsularis, the San Lucan banded gecko, and Coleonyx variegatus abaddii, the San Diego banded gecko, and along with Sleveni from Isla Santa Ynez, right? And then sister to that, let's say, was the variegatus group. Now this included the continental subspecies like Sonoriensis in Sonora, Bogardi in Arizona, widespread variegatus, and Utahensis in the northern Mojave. So again, Coleonyx variegatus being widespread throughout these warm desert regions, excellent species to kind of look at biogeographic models and the formation of Baja California. So kind of the questions I was asking with this study were, well, does the widely distributed Coleonyx variegatus, does it comprise one or multiple evolutionary lineages? Are Klauber's different subspecies distinct lineages? Are there morphologically cryptic or these undetected lineages that might be present within a widespread taxon like Coleonyx variegatus? Now, along with saying, are there different lineages, kind of more specifically, does the genetic structure within Coleonyx variegatus, does it fit Grismer's hypothesis of this peninsular group versus continental group, that these 2 groups formed with the formation of the northern Gulf of California? So I sampled geckos from throughout their distribution, included about 324 Coleonyx variegatus. 6 of the 7 subspecies were represented. The only one that was not was Sleveni from Isla Santa Ynez, which is in the Gulf near Mulegé. These data now might seem old, or it's a different kind of data set than is collected nowadays, perhaps. But my data set included 6 genetic markers, 2 mitochondrial, and then 4 nuclear loci. Essentially, all lizards were sequenced for the mitochondrial locus ND1, whereas a subset of those individuals were sequenced for the different nuclear loci. And I'm not going to go into the nitty-gritty of methods here. We'll kind of skip that part and jump to results. But basically, for the different genetic loci that I sequenced, I inferred species trees for those different loci individually. Additionally did some phylogenetic analyses and clustering analyses with combined data sets of those loci. So let's kind of just look at what happens when you sequence mitochondrial DNA from throughout the distribution of Coleonyx variegatus. Now, this is a mitochondrial tree inferred with a program called BEAST, which uses a molecular clock and Bayesian statistics to infer genetic relationships among, again, gene sequences, right? So there's a difference between looking at gene sequences versus phylogeny. They can relate, but this is looking at a gene sequence for the mitochondrial region. And there was some really deep structure within Coleonyx variegatus. You'll notice that there's a really divergent lineage, apparently sister to all other lineages from the Cape region in Baja California. There's a couple lineages in the northern Mojave, one in Inyo County that was unique, another one in northeastern Mojave that was unique. That kind of somewhat corresponds to the subspecies Utaiensis. There was a lineage in central Baja California. There's a lineage in coastal Sonora, and then there's a widespread lineage, right? So early on, I'm looking at these, wasn't quite these results here, but let's say an earlier version of these results, looking at these mitochondrial data, and I'm looking at these and I'm thinking, wow, there's like 6 different species found within Coleonyx variegatus. So as a reminder, for my master's program, I worked on Xanthusia vigilis group. And my work on Xanthusia vigilis, it was really following up this paper by Liz Sinclair, Bob Bezy, Jack Seitz, where it was kind of the first, I would say, comprehensive pass looking at the genus Xanthusia with DNA sequence data. And what they found with this paper and subsequently my paper is, well, when you find a unique mitochondrial lineage, you essentially have a unique species, right? So this paper and subsequent ones, they recognize Xanthusia gilberti from the Cape region as a different species, Extorus from central Mexico is a different species, the difference between Xanthusia arizonae, Xanthusia bezii, Xanthusia wigginsi, all of these things were more or less, let's say, recovered with the mitochondrial data, and they've held up through time with nuclear data. So coming from a Xanthusia perspective and seeing this, I was like, oh yeah, I've got 6 species. Well, we have known for some time that you can't trust mitochondrial data alone, even if it does a pretty good job with Xanthusia, right? And kind of getting to Grismer's hypothesis of Abadi group and a variegatus group. The Abadi group is here, kind of those branches are marked in red, the variegatus group are marked in blue, and these don't form 2 nice groups with the mitochondrial data, right? We have that Cape region population, which I can refer to maybe as Coleonyx variegatus peninsularis or the San Lucan banded gecko. Its sister is everything else. All right, yeah, it has this, maybe it has a vicariant origin with the formation of Baja California, but the rest of this Abadi group versus variegatus group didn't make a whole lot of sense with these mitochondrial data, right? So, you know, I didn't quite know what to think, but again, as collected nuclear data, I was able to analyze those and compare those with the mitochondrial data. So analyzing mitochondrial data phylogenetically is generally straightforward, let's say. We're analyzing a multi-locus nuclear data set. There are different ways to do it. You know, I did look at phylogenetic gene trees of the different loci. I'll mention one of those later. But otherwise, how to combine the nuclear data. The approach I took was essentially using a distance approach where I used a couple different clustering methods to see what are the different genetic clusters within Coleonyx variegatus, right? And kind of once I had an idea as far as what are these different genetic clusters, I could then go in with a phylogenetic approach using a program like StarBeast to look at phylogenetic relationships among these different groups inferred with these clustering approaches. So this network right here, this is a network where it's essentially it's analysis of the combined 4 nuclear loci. And what I'm doing here is essentially using this network approach to find what are these different genetic clusters. And you can see kind of up at the top on this long bunch of branches on this part of the network in purple is an outgroup to Coleonyx variegatus, the Texas banded gecko, Coleonyx brevis. And then these other 4 clusters from throughout the rest of the network correspond to Coleonyx variegatus. And essentially what I found were 4 different clusters, and these clusters held up with bootstrapping these data. So these clusters included kind of that San Lucan population in the Cape region of Baja California, included a cluster I'm calling the Abadai cluster or the San Diego banded gecko cluster, Sonoriensis or Sonoran banded gecko cluster, and then this widespread cluster, this variegatus cluster. Now, this cluster included, of course, the subspecies Coleonyx variegatus, but included other named subspecies like the Tucson banded gecko, Bogardi, and the Utah banded gecko, variegatus utaiensis. So it wasn't just this approach which kind of found these 4 different clusters. Using neighbor joining analysis and this same data set with the 4 different nuclear loci, again, I found these same 4 clusters. So the variegatus cluster, the Sonoriensis cluster, the peninsularis cluster, and the Abadai cluster. And these 4 clusters were found in 2 different groups, corresponding to Grismer's groups, the continental or variegatus group with Coleonyx variegatus sonoriensis, and this cluster that I'm calling here the Coleonyx the variegatus group as well. All right, so they formed a variegatus group or a continental cluster. And then there was an Abadai group cluster or a peninsular cluster, which is composed of the San Lucan banded gecko, Coleonyx variegatus peninsularis, and the San Diego banded gecko, Coleonyx variegatus abadai. So very different perspective than the mitochondrial data. I was surprised as this pattern emerged. And as you can see, 4 of these, well, 3 of these, let's say, correspond to named subspecies, more or less. And then the 4th one, variegatus, that's kind of more, does correspond to the desert banded gecko, Coleonyx variegatus, but it also includes those adjacent subspecies like Bogardi and utaiensis. So this is kind of what it looks like here, right? Again, our distance analyses do find multiple lineages within Coleonyx variegatus, 4 lineages, let's call them, and these 2 different groups. Knowing these clusters, then some of the patterns that I was seeing within the individual nuclear loci made a little bit more sense, let's say. Here's one gene tree of just one locus. I'm only going to show you one nuclear gene tree with this part of the talk. This is a gene called BAC1. And essentially with the nuclear loci, I found that 3 of the 4 loci found this peninsular group with the San Diego banded gecko and the San Lucan banded gecko, right? And the 4th one found those two separate groups, even if it didn't form a peninsular clade. So the individual nuclear genes were supporting individually kind of the big picture nuclear story as well. And if you look at kind of some of the details in this nuclear gene tree, you'll see, well, yeah, those clusters make sense, but sometimes you would find these exceptions, essentially sequences that are misplaced in the gene tree based on what their assumed placement according to the clustering analyses that I mentioned before. So for instance, I found there was a variegatus sequence that was grouping with the Sonoriensis individuals. And then, vice versa, found a Sonoriensis sequence grouping with variegatus individuals. Now, what was kind of clear that was going on with these individuals, that these misplaced nuclear alleles were the result of introgression. All right, so these different sequences were not grouping with the right cluster, but rather they were identical or nearly so to a neighboring lineage. These individuals, I could assume they were introgression due to their kind of phylogenetic position and their geographic position. Always, these misplaced individuals were right at boundaries between these different lineages. So what is introgression? Some of you folks, of course, know. Others, that might be a new term. Essentially, introgression is movement of genes from one species to another via recurrent hybridization, right? And we're all familiar with hybridization. Let's imagine two species, A and B. Their F1 hybrid would be 50% A, 50% B. But in subsequent generations, if that hybrid back crosses with only one of those parental populations, eventually that that hybrid will be brought into a single genetic cluster, let's say, with just traces of the other species present, right? So in short, there's numerous examples of this, some of which you've undoubtedly heard of before. One of the more kind of well-known examples of genetic introgression is between these two very different species, right? The North American bison and domestic cattle. We know the history of bison, that they were hunted almost to extinction, they went through this severe bottleneck, and that there were both intentional and accidental matings between cattle and bison. And due to that, eventually nowadays, bison herds, even though they look like bison, look very different from cattle, they do have some introgressed DNA from cattle. So they're not 100% what they were bison-wise from a few hundred years ago. They do have these traces of cattle ancestry. Okay, so to keep this story short, I did a number of tests to see whether or not these misplaced alleles were in fact introgression, and these tests did in fact indicate that yes, they were misplaced due to genetic introgression there at these boundaries between, say, Coleonyx variegatus and Coleonyx variegatus sonoriensis. And what happens is when you do use phylogenetic tools like StarBeast, this is a phylogenetic tool that is essentially modeling populations to infer phylogeny or evolutionary relationships among species. But this model does not include introgression or does not include gene flow among different species. And when you don't account for these misplaced alleles due to introgression, you end up with a different, in this case, the topology of the tree was the same, but the statistical support for relationships within this tree were lower when the introgressed alleles were present. And branch lengths within the phylogeny of introgressed alleles was compressed relative to a phylogeny where those misplaced alleles were kind of put in their donor gene pool, let's say. You don't need to know details of this, except the take home here is that yes, a variety of genetic tests do indicate that there was limited introgression there at the boundaries between these different lineages. All right, so getting back to the mitochondrial gene tree, I have that here on the left with those very diverse, divergent, very different lineages that maybe early on I was thinking was 6 different species. Nuclear data are telling me, no, doesn't look like 6 species. It looks like maybe 4 species or lineages. And one of the reasons why this mitochondrial tree was misleading is because there were a number of instances of mitochondrial introgression as well. There were a couple cases where there were individuals that belonged to the genetic cluster of Coleonyx variegatus abadi, the San Diego Banded Gecko up here, that were more misplaced, let's say, in the mitochondrial gene tree. There was an instance of that with members of the sonoriensis cluster also being misplaced. So again, the high similarity of these mitochondrial sequences, the geographic position, again, indicated that this was due to mitochondrial introgression. Mitochondrial introgression, some folks might see it as a nuisance. It can be if you're thinking about just inferring a phylogeny, but it also is informative thinking about genetic interactions among different species. And there's some really fascinating examples of mitochondrial introgression. You can see here again, this introgression happened a couple different places where these lineages were proximal, like in Sonora here. In yellow, we have members of the variegatus cluster. In blue is the sonoriensis cluster, and the introgression was due to those sonoriensis populations that were closest to the variegatus population. Similarly, those San Diego banded geckos, or the Coleonyx variegatus abadi, that had the wrong mitochondrial DNA, well, those were individuals from the northern peninsula that were proximate to these continental populations of Coleonyx variegatus. So there were numerous examples of recent mitochondrial introgression. Recent is in quotes because it's let's say Pleistocene, right? It might not be end of the last glacial maximum, but it's geologically young. All right. So once, so I mentioned a couple of the interesting examples of introgression. Some of my favorite have to do with a really cool lizard genus, the collared lizards, right? Crotophytus. So, for instance, throughout much of its distribution, the species down on the bottom, Crotophytus reticulatus, has the wrong mitochondrial haplotype. It has a haplotype it captured from the widespread Crotophytus choleratus, right? So that cool finding came out with a paper by Jim McGuire a while back, and kind of more relevant, I guess, to folks in Arizona. Two of the species there in Arizona, Crotophytus choleratus and Crotophytus bicunctoris, essentially all of the bicunctoris, Crotophytus bicunctoris populations there in Western Arizona don't have the original bicunctoris mitochondrial DNA. It's been captured repeatedly from the widespread choleratus species. So again, looking at mitochondrial data alone is misleading, but if you look at the whole picture, it shows kind of a cool story of maybe limited genetic exchange between these different species. So once I've identified those nuclear sequences that were introgressed, aka misplaced, those mitochondrial sequences that were introgressed and misplaced, I can infer a phylogeny for Coleonyx variegatus. And what happened, and trying to figure out, well, does this correspond to the formation of the Northern Gulf of California? It's a little bit tricky. Typically when you're creating a chronogram where you're trying to estimate divergent states, you want fossils to help you calibrate your molecular clock. Coleonyx as a group does not fossilize very well. They're small geckos. The bones are small. There's not a very extensive fossil record for Coleonyx. There's a bunch of late Pleistocene subfossils. A couple years back, there was a Coleonyx fossil actually discovered in Florida, of all places. No Coleonyx found in Florida now, but very limited. So what I did to infer divergence dates in a chronogram is I used a secondary calibration from a different gecko paper, right? So Cam Seiler had a gecko paper published a few years back. I used their mitochondrial substitution rate. And using that substitution rate, essentially I inferred a divergence state between the continental lineages, sonoriensis and variegatus. That divergence between the continental group and the peninsular group, comprised of the San Lucan banded gecko and the San Diego banded gecko, was around 6 to 6.5 million years ago. And lo and behold, that divergence state corresponds very nicely with geological data as far as when the Northern Gulf of California formed. There's been some really cool papers published over the past, I'd say over the past 10 years, but increasingly we have a better idea as far as when and how the Gulf of California formed. And some of these, one really nice paper I liked was by Scott Bennett published in 2015. And essentially this paper argued that the formation of the Northern Gulf from essentially the Santa Rosalia area in central Baja cleared up to, the southern area, southern Arizona, California area. It all flooded more or less the same time between 6 million and 6.5 million years ago, right? There's this narrow synchronous sea wave formed then, which again fits very well with our estimated divergence dates between our different lineages here. So to summarize kind of so far, yeah, 4 lineages within variegatus correspond more or less to 4 different subspecies. The continental group and the peninsular group of Grismer, we did recover that with phylogenetic data. And the timing, well, the timing is different than Grismer's hypothesis, but the timing of this divergence date is in keeping with what we now know from geology as far as the formation of a northern Gulf of California. So that's pretty cool. Along with that, there's some evidence of gene flow or introgression between adjacent lineages. So, one of the questions I wanted to know, well, I guess I'll get to that. One of the questions I wanted to know was, well, what happens where these different lineages come together? I ultimately focused on looking at an area in northeastern Baja California to address that. I would have liked to do that in California. We do see that the Coleonyx variegatus abadi, the San Diego Banded Gecko, and Coleonyx variegatus, the desert banded gecko, do come into close contact. And interestingly, these populations in yellow that I'm calling Coleonyx variegatus were, prior to this work, thought to be Coleonyx variegatus abadi, the San Diego Banded Gecko, because of general similarity in coloration. But genetic data say no, they're separate, and that Coleonyx variegatus abadi, the San Diego Banded Gecko, is restricted just to coastal San Diego County as far as United States goes. And it would have been nice to kind of look at hybridization between these 2 lineages here, but it's tough to sample that area really densely. So one of the outcomes of this study was that, with the understanding that the San Diego Banded Gecko abadi has a limited distribution in the United States, it was subsequently listed as a species of special concern. So there's a kind of conservation outcome from this work. Now, do these different lineages, I mentioned 4 lineages, do they retain their distinctiveness? This is a photo of Coleonyx variegatus abadi, the San Diego Banded Gecko. Now, Grismer, when he kind of talked about these different groups, the abadi group versus the variegatus group, yeah, he thought of them as being different, but he thought that where they came into contact, they essentially formed wide areas of intergradation, right? And I detected some hybridization or some introgression in northeastern Baja California. Now, this map here is from Grismer's Baja California Bible, let's say, and he shows this area near Cortecitos as being this wide intergrade zone. So I wanted to go kind of look at that more explicitly and go, well, what is happening here between these 2 lineages? Where 2 lineages come together, you essentially have 2 outcomes, right? One, these lineages that were at once distinct could come back together and merge, right? Or you could have hybridization, and these lineages, even with some hybridization, could retain their distinctiveness. So yeah, that's the general outcome. There's been some really cool hybrid zone studies looking at hybrid zones of fire-bellied toads in Europe. And essentially, I want to know, well, do these different lineages of geckos, do they retain their identity with a narrow hybrid zone where there's this balance between selection against hybrids and dispersal, or is there kind of neutral mixing, right? Are they just merging together? So, yeah, I want to know, are these things merging or maintaining their genetic uniqueness? So, if they were in fact merging, I would expect a wide hybrid zone, no selection against hybrids, A narrow hybrid zone would indicate that there's a barrier to gene flow between these lineages. So there would be selection against hybrids. So when you to look at this, I used Klein analyses, and these are some different examples of possible Klein shape. Kind of the upper left is a step Klein, where you're going from kind of one pure population to another pure population with a very steep Klein. Bottom right would be the opposite, where there's a very Kleinal variation is very gradual with no obvious Klein center. And then these other two Klein shapes are kind of in between, right? So top left is a more or less a steep Klein, but you can see there's some gene flow there at the edges, and then kind of the bottom left is more of a kind of tending towards a neutral Klein. All right, so if it's a neutral mixing, I expect this wide contact zone where essentially the different genes would have different Klein centers. They would have non-concordant Klein widths. If there was selection against hybrids and there was essentially they maintained their identity, the Klein center would be coincident, and then the Klein widths would be concordantly narrow. So I did sample the transect essentially between San Felipe and Puertecitos. In that transect, collected tissues from around 71 geckos, and included geckos from outside of that hybrid zone as well. I used 6 different loci, 5 nuclear loci, and mitochondrial locus, used Klein analyses using this program KleinFit, and then I calculated hybrid indices. So long story short, or we'll cut to it really fast, when you're driving along this road, sampling geckos from San Felipe to Puertecitos, there is a very marked shift in the morphology of geckos from variegatus-like to abidae-like, about 31 kilometers north of Puertecitos. And that these variegatus-like geckos, desert banded geckos, are much more abundant than the smaller San Diego banded gecko, at least encountered 5 times more frequently, even when trying to focus on collecting the San Diego banded gecko. So morphologically, it was not a broad zone of intergradation. It was pretty abrupt. And looking at the genetic loci, this is looking at the 5 nuclear loci. Well, lo and behold, that is the genetic transition is reflected in this shift 31 kilometers north of Puertecitos as well. Yeah, there's some introgression between these 2 lineages, and it's bidirectional, right? There's bidirectional gene flow, but generally it's bidirectional, but kind of asymmetric, with more abidae alleles flowing into variegatus. So the morphological transition equaled the genetic transition. And the KleinFit model was, that these 6 loci show a coincident Klein center. They're all at the same place. And essentially the 6 different loci fell into 2 categories. Some had very narrow Klein widths, others were slightly wider. So there was really high concordance, even if it was incomplete. Okay, this is looking at the hybrid index, where essentially an index of 1 is pure variegatus, an index of 0 is pure abidae, and you see this very abrupt transition from abidae-like animals to variegatus-like animals there north of Puertecitos. So a narrow Klein shape, coincident Klein center indicated, there's a strong barrier to gene flow. Like I mentioned, this gene flow was asymmetric. That's best explained by hybrid zone movement. And the scenario there would be that the continental desert banded gecko expanded its distribution to the south at the expense of the San Diego banded gecko, and that the higher population density of the desert banded gecko is really the mechanism that allows it to kind of push out the San Diego banded gecko there in northeastern Baja California. So this is just kind of another way to look at it. In green are the San Diego banded geckos. In orange are the desert banded geckos. Now this Klein center is not near any sort of ecological ecotone or climatic ecotone, but there is at least a subtle shift here from kind of really sandy areas to the north to kind of more rocky volcanic alluvium to the south. So there might be some environmental story at play here, even if it's not vegetation or climate. All right, so conclusions of the hybrid zones. Yeah, it's consistent with hybrid zone movement. There's introgression between the two, but selection against hybrids. So the take home now, I mean, for me is like, well, are these different subspecies or different species? I would say that, you know, I'm honestly fine with either, but gene flow is limited enough between these two lineages that I prefer thinking of Coleonyx variegatus abadi, the San Diego Mesa gecko, as its own distinct species, Coleonyx abadi. Now, I know kind of running low on time, but wanted to mention really fast. Well, what's up with the Tucson banded gecko, right? Our mascot for THS. Well, the Tucson banded gecko is, you know, it's characterized by these kind of wide bands with a kind of double dark bar. They have a wider mental scale on their chin. And then kind of the character that really sets them apart is the males typically have 8 or more preanal scales, where the femoral pores, where other populations do not, right? So, you know, one outcome of my work was that maybe the Tucson banded gecko was synonymized with Coleonyx variegatus. And we hold on to the Tucson banded gecko. So here's a distribution from Stebbins from a few years back. This graph right here is something that Bob Bezy put together, looking at these distinctive counts of male bogardi, showing that there is there is distinctiveness here with bogardi versus kind of the widespread variegatus. With genetic data, there's something going on. The distribution of bogardi essentially corresponds to a subclade mitochondrially. And looking at the nuclear data, so there it is right here, that Eastern Arizona subclade does kind of correspond to the distribution of bogardi. And looking at the combined nuclear data with this neighbor joining tree, well, they do cluster together with some proximal individuals from like Maricopa County nearby as well. So, and then other, you know, Arizona geckos from say La Paz County or Mojave County, or, you know, some of them from Yuma County were outside of that little cluster. So can we save the Tucson banded gecko? Well, I would say if you want to save the Tucson banded gecko, you would take approach similar to Dustin Wood's paper, where argued for maintaining the Tucson shovel-nosed snake, right, clobberide. So this paper, fuzzy boundaries, color and gene flow patterns among parapatric lineage of the Western shovel-nosed snake. Well, essentially, Wood et al. Came up with a taxonomic arrangement that essentially retained clobberide, the Tucson shovel-nosed snake, as distinctive, with maybe a slightly wider distribution than was previously thought. I would think that a similar approach could be taken with Coleonyx variegatus. So a possible taxonomy for Coleonyx variegatus is, well, you could elevate these different lineages that I found in my work. Coleonyx abadi would be the San Diego banded gecko. Peninsularis would be the San Lucan banded gecko. Sonoriensis would be the Sonoran banded gecko. And then within the Western banded gecko, Coleonyx variegatus, you could retain these subspecies like bogardi or Utahensis, where there is some population substructure where that corresponds with those named subspecies as done by clobbering. So there is a way to do it if you took this approach. So as far as like looking at genetic substructure within Coleonyx variegatus, I think some of the remaining big questions that need to be asked as well. I looked at that hybrid zone between Coleonyx abadi and variegatus. What's happening where the different lineages come together in Sonora? All right, so characterizing the Sonoriensis variegatus contact zone is really relevant. I think it's relevant if you want to, you know, come up with a taxonomic arrangement like this, right, where Sonoriensis is a distinct species. I would like to characterize the contact zone between abadi and Peninsularis in Baja California. And then, you know, there's populations in Southern Sonora, Northern Sinaloa, where, you know, we don't really know what's going on with them genetically, and including them with these data sets would also be useful. All right, so Anyway, I know that's a rush through kind of my work. I'd like to thank the folks that have been collaborators with me, Brad Hollingsworth, Todd Reeder, Robert Fisher. I'd like to folks others that have been really generous with their time and samples. Bob Bezy, many of my Arizona Coleonyx came from him. Peter Scott was a lot of help in the field. Annie Peralta Garcia and Jorge Valdez helped a lot with some of the Baja and Sonoran samples. Dustin Wood from the USGS helped a lot. My wife went out in the field a few times and helped collect geckos. And there were a whole lot of others that helped with various components of this study. So I guess with that framework, I'll just end with, are there any questions you folks would like to ask about banded geckos and what I presented?
I have a very important question. Which is the cutest subspecies?
What is the cutest subspecies? Depends on if they're adults or hatchlings. I would say that the San Diego banded geckos, it's pretty cute because it's smaller. It's small and pretty adorable. But then I really like the pattern of the San Lucan banded gecko. It's my favorite color pattern. So
It's the Tucson banded gecko.
I failed. They're pretty adorable.
I'm gonna if you could stop the screen share so we can get to the chat questions. Thanks.
Yeah, so there's a question like with the integrate regions, how can you tell whether an individual is hybridized or not? My approach is just doing that genetically, right? Like, so there's, with the contact zone that I looked at more extensively in northeastern Baja California, well, if it's if there's introgression, you would just detect that genetically. And you couldn't necessarily detect that morphologically. There were individuals that looked, like they look just like Abidae that might have one of their alleles be variegatus, and conversely, something that's very desert banded gecko-like but have an Abidae allele. So, those things have crossed back to the parental populations enough where the overall morphology, the overall anatomy of these things is very much like kind of the quote unquote pure ones. It's really only right near the Klein center where you might get something that looks kind of in between. And there weren't really any obvious individuals there that were right in between. Maybe part of that's because a couple of those geckos had been run over a couple times by a car. So I was scraping up DNA and really couldn't look at the morphology that well. But there wasn't a lot of intermediates, to say the least. I didn't see any obvious intermediates.
Question.
Dean, I am unmuting. Can you hear me okay?
I can hear you.
Great. That was a fantastic talk. Really enjoyed it. I was struck by the narrowness of the hybrid zone, and I'll call it that because to my mind, they're different species there, what you described in Mexico. The way they're behaving reminds me of the whiptail hybrid zone in eastern Arizona between Marmoritas and Tigris. But I actually had a question for you that's completely a natural history tidbit. You mentioned the higher densities of the one to the north, variegatus, and lower densities of Abidae to the south. So you were typically night driving to pick up the geckos off the road. And I'm just curious if you have an explanation for this or saw this in your other efforts around the Southwest. So the all-time record I have for encountering variegatus, and that's just here in central Arizona, south of Phoenix on a road that we used to survey for toads, was I found 50 in about 40 minutes in about a 5-mile stretch. And it was just as the sun set. There had been a monsoon rain in the early afternoon, and I'd never experienced anything like that. And I don't know what the numbers are per kilometer or whatever, but 50 in 40 minutes was just an astronomical number. I'm curious if you've ever encountered something like that.
I haven't ever seen it with that high of number. I've definitely seen, let's say 30 plus in a good night in kind of the Western Imperial County. So, a May, yeah, essentially typically a good May night, you can see a whole lot of them in the desert part of Southern California. So that, I've had good gecko nights with variegatus. There's nothing, I've never seen numbers, anything like that at all ever with the populations in Baja California. They're much harder to come. So the population is just much less dense, Coleonyx variegatus wise throughout Baja California. It's not just that that hybrid zone there in northeastern Baja California. So Abidae and Peninsularis, they're much harder to collect than variegatus.
Cool, thank you.
Thank you. We have a question here.
Yeah, with the introgression, is that accelerating due to climate change, or is there any work being done on that currently?
No, that's the big question. I mean, that is my big question, right? And I would hypothesize that that's likely. I would love to revisit that to see if there has been a hybrid zone shift. But yeah, I would expect that that's the case, that variegatus are better adapted to warmer climates, and that they will continue to kind of push south at the expense of Abidae. Now, based on like genetic diversity mitochondrially with those two groups, I think that that hybrid zone it's fairly old. It's not post-Pleistocene or anything like that. It's older than that. So Abidae have been kind of holding on there for some time. But I know with like looking at hybrid zone movements with different bird species, that climate change will accelerate hybrid zone movement. And I think that's I don't know if it's definitely a possibility with this system as well.
Thank you very much.
So I've got a question.
Yes, I've forgotten. It's interesting that you find no difference between true San Lucan Cape region geckos and those in the central part of the peninsula. Do you feel confident that you have enough data there, or what do you feel about it?
Well, there's definitely a sampling gap between what I'm calling Abidae and the San Lucan Banded Gecko. So where the essentially I have all the individuals I have for the San Diego Mesa Gecko, they're kind of, they get to where the contact zone is supposed to be, but it's how should I say that? To characterize contact between San Lucan Banded Geckos and San Diego Mesa Geckos, it's not just looking at the hybrid zone, it's actually finding where that contact is. And I'm not confident that I know where that is right now to even do a hybrid zone type of thing. Sampling in that region is a little bit challenging.
Yeah, but say within Peninsularis, you haven't picked up any differences between actual Cape region population and central peninsula population.
Oh, the stuff to the north of the isthmus. There isn't a big break isthmus wise. There is with mitochondrial data. With mitochondrial data, there is some difference there, but with nuclear data, there's not.
That's it.
Yeah, I understand your question now. Yeah, so mitochondrially, it's actually quite divergent, but the nuclear data, there's gene flow between there.
Thanks.
And there's a question in the chat as far as in the hybrid zones, do you have an idea of what percentage of the genome is introgressed? Any plans of some kind of phylogenomic analysis to identify loci that are barriers to introgression? That would be kind of a next step. I'm not currently personally doing that. I am collaborating with someone that's doing a subgenomic kind of follow-up to this study using what are known as DD-RAD data, where you're looking at more of the genome at once, but that is not looking at the hybrid zone per se. So that's definitely something that I think would be really cool. I think, I have samples to do that, and there's tools to do that. But yeah, what are there specific loci involved with this variegated gene flow? Is it kind of, are there loci that have, crossed this this hybrid zone and have spread due to positive selection? Those are questions that have yet to be addressed.
Dean, we have a question here in the audience.
Okay.
Okay, so if I could ask a couple of questions about the Coleonyx fossils found in Florida. So which part of Florida were they found? Morphologically, how do we know they belong to Coleonyx? What museum are they deposited? And what's the full species name of that material?
So I can't answer all of those. If my recollection is correct, they're at the University of Florida. It's in a dissertation. There's not a name ascribed to those fossil species, to that fossil material. And I can't remember exactly. I'm trying to remember the name of the local flora that it was part of. I'm blanking right now. If you're interested, you could message me and I could find the dissertation that discusses that. But
Is it a subfossil or an actual fossil?
no, this is fossil. This is like early Miocene or Oligocene fossil.
And tagging on to the fossil theme, did you look at like pack rat midden analyses or like sort of Pleistocene floras and how they might correspond to what you're observing?
So that's one thing I was really interested in kind of early on that I thought about talking about, but I haven't published on. And that kind of relates to what's known as the Pleistocene stability model, at least in part. And that model is more or less as, kind of modern reptile amphibian faunas are more or less unchanged through the Pleistocene. Now there's some exceptions for that. Cutting to the chase, one thing that the genetic data, the mitochondrial data from this study shows is that Coleonyx has persisted in their current distribution through most of the Pleistocene. There's only a couple small places where there's evidence of post Pleistocene range expansion, like Inyo County, maybe easternmost Arizona, coastal California. But everywhere else throughout core California, throughout core Arizona, even in southwestern Utah, branch lengths of those mitochondrial data indicate that those geckos persisted in situ throughout the Pleistocene, which means that the pack rat midden data where they do find Coleonyx remains, well, it is consistent with that, right? So I mean, it's an interesting question, which lizards kind of stood in place throughout the ice ages and which ones had major, range contractions and range expansions, right? Kind of as opposed to these geckos, which more or less stayed there based on the genetic data. You have things like, the desert iguana, really a heliothermic lizard, likes it hot, and the genetic data say the opposite, right? That they had a much more restricted distribution during, the last glacial maximum, and that their current distribution is a result of a huge range expansion, right? So I would say just generally the pack rat midden data combined with this data are agree that geckos persisted throughout much of their core distribution throughout the ice ages.
I forget which subspecies you were making an example of between sandy and rocky habitats.
Well, so in northeastern Baja California, variegatus, the desert banded gecko, they're really in these sandy, duny areas where abidae, kind of you run into them where it started to get more this rocky alluvium, right? So those were the two that were kind of interacting there. In Imperial County, the desert banded gecko, variegatus, they love duny areas. But then I know, elsewhere in Arizona, variegatus and bogarti, they get to be in some pretty, they like boulder fields as well.
Well, I asked because I know the sand is also very recent. It's like within the last not even 10,000 years. It's like 7 to 2,000 years that all of that has accumulated up on the mainland.
Right, so they do great in sand. They're not like restricted to that windblown sand. But another kind of question that ties into that, I guess, is, you know, that contact zone I was looking at, you know, that road is pretty close to the coast. You know, you think about, and it's the genetic data indicate it's old. Well, what was going on, you know, during the last glacial maximum and when the sea level was lower, right? Was there kind of a greater area for those two different lineages to kind of interact genetically? Yeah, maybe. So there are kind of some interesting questions there as far as how substrate and kind of sand through time is influencing their genetic interaction.
Brian has a question.
Yeah, so if I'm not keeping others that are there in person from asking, I'll follow up on that. I was struck by how the distribution from San Diego to the south, Abaddon, it in essence crosses over from what's a coastal gecko to being one that's in some of the, you know, harsher desert environment there on the Sea of Cortez. And likewise, variegatus, if it is punching through to the coast north of San Diego. So those historically are the notions of subspecies. You're using color pattern and habitat, and yet those two suggest there may not be the hard-wired connection between those two, if I'm making sense. Have you given much thought to that?
So, right, there's kind of an idea that the San Diego banded gecko, they kind of have an overall darker coloration than the desert banded gecko, and it might be this coastal adaptation for cooler temperatures, right? And so then kind of if you have essentially what's a desert banded gecko punch through like San Gorgonio Pass and kind of head towards the coast, did they also, is their kind of Abaddon-like pattern, is that a result of convergent evolution? Yeah, I've thought about whether it's convergent evolution or whether there's even the possibility of introgression between those two, influencing those coastal desert banded, the coastal desert banded geckos, whether that has played some role. And it, you know, you could ask those questions and look at that with like a subgenomic or genomic data set, but it's an interesting question. Interesting, you kind of mentioned, you know, the Abaddon or the San Diego banded gecko being a coastal thing for much of its distribution, and you get kind of south of the, you know, get into the Valle de los Cirios and down to Vizcaíno, and it's less so, right? Well, the pattern of Abaddon kind of throughout the central Baja California is much, there's a lot more variation in pattern. It doesn't have that very, it doesn't always have that very clean pattern that coastal San Diego banded geckos have. So if you look at geckos from like Valle de los Angeles or, you know, Vizcaíno region or kind of near San Ignacio, you know, they might look kind of Abaddon-like, like the ones from, you know, San Diego County, or they might have a more broken up pattern. It's less, yeah, there's more variation there.
Cool, thanks very much.
I think for the sake of time, we're going to have to end the meeting, but thank you again for presenting, Dean, and thanks for everyone for all the great questions.
Thanks, folks.